Hello everyone, and thank you so much for joining today's webinar. My name is Naja Abijarjes, and I am a Senior Vice President of Research Alliances at Anabios. Today, I'm delighted to share with you how Anabios is helping pharmaceutical and biotechnology companies around the world, including companies in South Korea, generate human predictive data before entering clinical trials. The title of today's presentation is Human Predictive Data Before the Clinic. Over the next 40 minutes, I will walk you through our science, our validated models, and how they can directly support your drug development programs. Please feel free to write down any questions, and we will address them at the end. Let me begin by briefly introducing Anabios. Anabios is headquartered here in San Diego, California, one of the world's leading hubs for biomedical research. We have been generating human predictive data for 15 years. In that time, we have worked with more than 200 pharmaceutical and biotechnology partners worldwide, helping them to de-risk their pipelines. More than 50 of the drug programs we have supported have successfully advanced to IND application with the US Food and Drug Administration, the FDA. And we are proud to serve as a research partner with the FDA itself, working directly with regulators to advance the science of human tissue-based testing. These numbers reflect 15 years of focused commitment to one mission, providing pharmaceutical companies with the human data they can trust before going into patients. Now let's talk about the core problems that Anabios was founded to solve. Drug development today suffers from a very serious translational gap. What does that mean? It means that data generated in animal models, mice, rats, dogs, rabbits, very often does not predict what will happen when the same drug is given to a human being. The consequences of this gap are dramatic. Approximately 90% of all drugs that enter clinical trials ultimately fail. This is not because scientists are not working hard. It is because the models they are using, i.e. the animal models, are fundamentally different from human biology. As you can see on this slide, when a drug is tested in animal models and then moved into clinical trials, the result is very often a clinical failure. But when drugs are first tested in human tissue before entering clinical trials, the probability of a clinical success improves dramatically. This is the core value proposition of Anabios. We help you predict the clinical outcomes, not just animal responses. And this is not just a scientific argument, it is also a business argument. Every clinical failure costs tens or hundreds of millions of dollars and can cost patients their safety. So how does Anabios actually deliver on this promise? It all starts with tissue quality. At Anabios, we believe that you can't generate reliable, reproducible scientific data if you start with poor quality tissue. The quality of the data is entirely dependent on the quality of the starting material. Our process has 3 critical steps. First step, we collect human organs from donors and preserve them specifically to maintain viability. Every decision in procurement is made with the goal of keeping the cells alive and functional. Second step, the organs are shipped to our lab and then quality controlled and validated before any experiment begins. We do not use tissue that does not pass our quality standards. Third steps, we generate functional data. Data that reflects how living human cells actually respond to drugs. This is the kind of data you can take to regulators. Why does our tissue perform differently from what you might find elsewhere? Because we have consistent results across multiple donors, because our cells are highly viable, meaning they are alive and functioning, because we operate a large network, giving us access to a high volume of organs, and because we maintain full donor history for packages to be submitted to regulators. This infrastructure is what makes our data trustworthy. Anabios works across multiple organ systems and disease areas. Let me give you an overview. We work with 6 major tissue types: peripheral nerves, lungs, the heart, kidneys, the colon, and the skin. For each of these tissues, we can address both efficacy and safety questions. On the efficacy side, for example, we study pain using peripheral nerve tissue. We study lung diseases such as fibrosis, COPD, and asthma. We study heart disease, including atrial fibrillation and heart failure. And we study kidney disease, gastrointestinal conditions, and itch. On the safety side, we have validated assays for neurotoxicity using peripheral nerve tissue and cardiotoxicity using heart tissue. These are the 2 most common safety liabilities that cause drugs to fail or be withdrawn from the market. Today's webinar will focus primarily on our cardiac and lung models. Before we dive into our functional heart and lung models, I want to show you one key data point that illustrates the quality difference of anabios tissue. This chart shows what is called RNA integrity number, or RIN score. It is a standard measurement of how well preserved the RNA in a tissue sample is. A score of 8 or above is generally considered the minimum threshold for reliable RNA sequencing. You can see that across multiple tissue types, heart, dorsal root ganglia, spinal cord, kidney, lung, and liver, anabios tissue consistently achieves RIN scores well above 8. In many cases, we achieve scores of 9 or even 10. The competitor tissue shown in gray consistently falls below the threshold of 8 in several tissue types. Why does this matter? It matters because high RNA integrity enables robust transcriptomics studies, clean proteomics, and reproducible immunohistochemistry. In simple terms, if you want to understand whether a drug target is actually expressed in human tissue or whether a drug is changing gene expression, you need high-quality RNA, and anabios tissue delivers that. This is not just a technical detail. It directly affects the confidence in target validation studies that are critical in early drug discovery. Now let me frame the broader value of human tissue studies with 4 fundamental questions that every drug developer must answer before committing to a clinical trial. Question 1. Is the target relevant in humans? Many drug targets that look promising in animal models turn out to be expressed differently or not at all in the relevant human tissue. Question 2. Does the compound work as expected in humans? Even if the target is relevant, the drug's mechanism of action may behave very differently in human cells compared to animal cells. Question number 3. How does the compound compare with the current standard of care? Before entering clinical trials, you need to understand your competitive profile. Question 4. What is the right dose for first-in-human studies? Getting the dose wrong can be dangerous for patients and devastating for a clinical program. The powerful message here is this: human tissue studies can answer all 4 of these questions before you commit the significant resources required for clinical trials. This is what we mean when we talk about de-risking clinical development. Now let's focus on our cardiac models. There are 2 fundamental questions drug developers ask about the heart, and they lead to 2 different paths. The first path is the safety path. The question here is my compound safe for the heart? This is critical for any drug, not just cardiac drugs. Many drugs developed for other diseases can unexpectedly affect the heart. In our safety path, we use a human heart tissue to detect arrhythmia risk, QT prolongation risk, conduction abnormalities. We do this before your compound ever enters a clinical trial. The second path is the disease path. The question here is, will my therapy actually work in diseased hearts? This is relevant for companies developing treatments for conditions like heart failure or atrial fibrillation. In our disease path, we use tissue from donors who actually had the disease, hearts with heart failure, or hearts from patients with documented atrial fibrillation, because normal heart tissue will not give you an accurate picture of how your therapy will work in a patient's diseased heart. Both paths lead to the same goal: human answers before the clinic. Let's start with cardiac safety. The risk here is significant. Nearly 30% of all drug withdrawals from the market are due to cardiac safety failure. These are drugs that appeared safe in preclinical testing using animal models and human iPS-derived cardiomyocyte, but caused dangerous heart problems when given to patients. One major reason for these failures is that human iPS-derived cardiomyocyte, which are stem cell-derived heart cells, produce false negative results on known dangerous compounds. In other words, these cells models fail to detect the danger. The solution is adult human tissue. Adult human cardiomyocyte, cells isolated directly from donor hearts, express the full complement of ion channels that are present in a real human heart. This is something that iPS-derived cells simply do not do. Anabios has validated 4 specific cardiac safety models. The first model is our CardioPrime assay, uses adult human cardiomyocyte, and is validated for both arrhythmia screening and contractility effects. Second model is our ProArrhythmia assay that uses ventricular trabecula, strips of muscles from the heart, to detect the QT prolongation early after depolarization and the triangulation. These are the electrical signatures of dangerous arrhythmias. Third model, we have a contractility assay, also using ventricular trabecula to detect the risk of contractile impairment. And fourth model is our conduction assay using Purkinje fibers, the heart's natural specialized conduction system to measure effect on action potential propagation. The bottom line, do not defer the cardiac safety question to a $50 million phase 2 clinical trial. Answer it now in human tissue. Let me describe our flagship cardiac model in more detail, the CardioPrime. CardioPrime uses freshly isolated adult human cardiomyocyte, single heart cells taken directly from donor hearts. These cells are not stem cell-derived cells. These cardiomyocytes are real, mature human heart cells with all the ion channels, signaling proteins, and contractile machinery of a functioning adult heart. What makes CardioPrime unique is that it detects two cardiac risks simultaneously in a single assay. Most assays can only detect one risk. First, CardioPrime detects contractility effects, whether a drug reduces or increases the strength of each heartbeat. This is measured in real time from multiple cells simultaneously. Second, CardioPrime detects arrhythmic risk, including transient arrhythmias that endpoint-based assay simply can't capture because they only measure at a single time as a single point in time. CardioPrime also provides a full dose response curve, allowing you to define the therapeutic window, i.e. the margin between efficacy and toxicity for your compound. This methodology has been published in peer-reviewed scientific journals, including the Journal of Visualized Experiment JOVE, Nature Scientific Reports, and Frontiers in Physiology. Our data is reproducible and scientifically credible. I want to address a question that many drug developers have. Why not just use human iPS-derived cardiomyocyte? They are commercially available, they are scalable, and they are less complex to work with. So why use primary human cardiomyocyte? The answer is on this slide. Human iPS cardiomyocyte are not the same as adult cardiomyocyte. They are not a good model of the adult human heart. On the left, you see a molecular analysis, a PCA of gene expression. Human tissue from donor hearts cluster tightly together with anabios CardioPrime cells. They have similar molecular profile. Human iPS cardiomyocyte, both monolayer and engineered formats, cluster in a completely different part of the plot. They are distinct molecular profile. On the right, you see the structural differences. Adult human cardiomyocyte are rod-shaped and show clear striations, the organized sarcomere structure that is essential for proper contractile function. iPS cardiomyocyte are round and disorganized. They look like fetal heart cells. They are not adult heart cells. And here is the critical point. Non-physiological morphology leads to non-physiological drug responses. If the cell does not look like an adult cardiomyocyte, it will not respond to drugs the way an adult cardiomyocyte does. When you are making go, no-go decisions that affect your entire drug development program, you need data from models that actually match native cardiac tissue. Now let me show you the clinical evidence for why human primary cardiomyocyte outperform human iPS-derived cardiomyocyte models. In a peer-reviewed study published in Frontiers in Physiology in 2017, our team tested a panel of 25 drugs with known clinical cardiac risk profiles, drugs that were withdrawn or restricted due to cardiotoxicity. Our CardioPrime model, using a human primary cardiomyocyte, detected 24 out of 25 of these dangerous drugs correctly. That is a sensitivity of 96%. But here is what is alarming. When the same drugs were tested in human iPS-derived cardiomyocyte models, the models, for example, failed to detect 5 of the most dangerous drugs. Look at the table on the right side of this slide. Drugs like cisapride, astemizole, terfenadine, pepridil, and chlorpromazine were all identified as proarrhythmic by human primary cardiomyocyte, but human iPS-derived cardiomyocyte models gave a false negative on all 5 drugs. These are not obscure compounds. These are drugs that were actually on the market and had to be withdrawn because they caused cardiotoxicity in patients. If these compounds had been tested with human iPS-derived cardiomyocyte models in development, they might have advanced to the clinic and potentially caused more harm. Human primary cardiomyocyte catch the danger. Human iPS-derived cardiomyocyte models do not. Now let me address the other side of the equation, false positives. A false positive occurs when a test incorrectly flags a safe compound as dangerous. This is a serious problem in drug development because it can cause companies to abandon promising drugs that are actually safe and effective. In the same validation study, we also tested 10 drugs that are known to be clinically safe, drugs that are approved and used routinely in patients today. Our human primary cardiomyocyte model correctly identified all 10 of these drugs as safe, 0 false positives, 100% specificity. Now look at what happened with the human iPS-derived cardiomyocyte models. Look at the table. Drugs like ranolazine, verapamil, mexiletine, diltiazem, and diphenhydramine, all safe, approved drugs, were flagged as positive by human iPS-derived cardiomyocyte models. These are false positives. If these drugs had been in development and tested only in human iPS models, the false positive signal could have led companies to abandon them, wasting years of research and millions of dollars, and ultimately denying patients access to beneficial medicines. Human primary cardiomyocyte give you the truth. False positive waste time and money. And in this industry, time and money are everything. Let me briefly summarize what we have just covered. Human primary cardiomyocyte achieve 96% sensitivity, meaning we catch almost all dangerous drugs that would otherwise proceed to the clinic. And 100% specificity, meaning we do not generate false alarms that would kill promising compounds. Together, these two numbers mean 3 things for your drug program. First, you will not advance a dangerous drug into the clinic. Second, you will not kill a good compound based on unreliable model data. And third, your cardiac safety data will be aligned with ICH S7B, the international guideline for cardiac safety testing. Human tissue gives you accurate data before it is too late. In addition to our cardiomyocyte model, Anabios has developed and validated a model using ventricular trabecula, small strips of a human heart muscle tissue. Trabecula are prepared from viable donor hearts, typically within hours of procurement. So the tissue is alive and fully functional when experiments begin. In this model, we look for a specific electrical signal called an early after depolarization, or EAD. An EAD is the electrical marker of arrhythmia risk. It tells us that the drug is disrupting the normal electrical pattern of the heart in a way that could cause dangerous rhythm problem in patients. You can see an example on this slide. The red trace shows what happens when a known arrhythmic drug, dofetilide, is applied to the trabecula. The early after depolarization event is clearly visible. The black trace shows normal behavior in the vehicle control. This model has several key performance characteristics that make it particularly very valuable for drug development. First, it predicts QT prolongation and arrhythmia risk with high accuracy. Second, it is highly reproducible. Variability across donors is only 7%. And third, it has been validated against 33 drugs with known clinical outcomes. The reference for this work is Page et al., published in the Journal of Pharmacological and Toxicological Method in 2016. I want to highlight something that is very important for your IND strategy. FDA scientists themselves, in a peer-reviewed publication in Frontiers in Pharmacology in 2025, concluded that the human primary tissue data correlates with the clinical ECG outcomes and enables predictions of drug-induced arrhythmia risk. This is a significant statement. The agency is telling the scientific community that the human primary tissue is a better predictor of clinical outcomes than human iPS-derived cardiomyocyte models. The implication for your IND submission is this. INDs that rely only on human iPS-derived cardiomyocyte data are facing increasing scrutiny from reviewers who understand the limitation of these models, including their tendency to produce false negatives and their immature fetal-like phenotype. On the other hand, including human primary tissue data in your cardiac safety package strengthens your submission and demonstrates translational relevance, a direct link between your preclinical data and what is likely to happen in human patients. In short, align your cardiac safety strategy with FDA-owned published science, and Abios can help you do exactly that. Now let me shift from cardiac safety to cardiac efficacy. If you are developing a therapy for heart failure or atrial fibrillation, you need to test your compound in tissue that actually reflects the disease you are treating. Diseased hearts are fundamentally different from healthy hearts. They have altered receptor expression, they have different ion channel profiles, and they have impaired calcium handling. A drug that shows positive result in a healthy heart may behave very differently in a diseased heart, and vice versa. Anabios has 3 validated cardiac disease models. First, our HFrEF model. HFrEF means heart failure with reduced ejection fraction. This is the most common form of systolic heart failure. Our model uses tissue from donors with documented HFrEF, and it has been used to validate the preclinical activity of APD-418, a compound that has received FDA Fast Track designation, and it has been developed by Arena Pharmaceutical and Pfizer. Second model is our HFpEF model. HFpEF means heart failure with preserved ejection fraction. This form of heart failure affects diastolic function, i.e. the heart's ability to relax and fill with blood. It is a growing area of unmet medical need with very few approved therapies. Third model is our atrial fibrillation model, using tissue from donors with documented atrial fibrillation. Atrial fibrillation tissue undergoes a specific electrical remodeling that create the substrate for the arrhythmia. Testing in atrial fibrillation tissue has already supported the development of a compound that filed IND. The message here is clear. Get a human efficacy proof of concept in your target disease before committing to the clinic. Let me share a specific example of how our atrial fibrillation tissue model works. This data comes from a 2021 publication by unbiased scientists studying a compound called GS967. This compound is a late sodium channel inhibitor being investigated for atrial fibrillation. In this experiment, we used atrial trabecula from donors with confirmed atrial fibrillation. In atrial fibrillation tissue, the heart generates not only normal stimulated contractions, but also spontaneous abnormal contractions called after contractions and premature atrial contractions. These abnormal contractions are what drives the arrhythmia in patient. Panels A and C on this slide show the atrial fibrillation tissue with vehicle control, meaning no drug. You can see irregular patterns of contraction with multiple types of abnormal beats. Panels B and D show the same tissue after treatment with GS967. The after contractions disappear. The contraction pattern becomes regular and organized. And panels E and F summarize the data from multiple tissue. GS967 significantly reduced both the amplitude of irregular contractions and the incidence of after contractions by approximately 50 to 80%. This is exactly the type of a human efficacy proof of concept data that gives confidence before entering a clinical trial, and it can only be obtained in tissue that actually has the disease. Here is another example, this time with a novel ion channel inhibitor that we call drug X, a compound developed by one of our partners. Again, we use atrial trabecula from atrial fibrillation donors. In the left panel, the vehicle control, you can see the hallmarks of atrial fibrillation tissue, irregular contractions, after contractions marked by the black arrows, and even triggered contractions marked in red. After treatment with drug X at a concentration of 0.1 micromole, shown in the right panel, all of these abnormal events disappear. The contraction pattern becomes completely regular and clean. This is a very clear and compelling efficacy signal. It demonstrates that the drug X is capable of suppressing the electrical and mechanical abnormalities that are characteristic of atrial fibrillation in human atrial fibrillation tissue. This is a human data in a human disease model supporting the IND submission. One of the most important safety questions for any atrial fibrillation drug is, does it affect the ventricles, the main pumping chambers of the heart? An ideal anti-atrial fibrillation compound should work selectively in the atria, the upper chambers of the heart, without affecting the ventricles. If it affects the ventricles, it may cause ventricular arrhythmias, which are far more dangerous than atrial fibrillation. This is called atrial selectivity, and it is a critical requirement for atrial fibrillation drugs. In this study, we tested drug X in two ventricular tissue models. isolated human ventricular cardiomyocyte on the left and human ventricular trabecula on the right. The left panel shows contractility measurements across a range of drug X concentrations. Notice that drug X shown in the colored lines does not significantly change the contractility profile compared to the vehicle control in black. This is in contrast to ISO, a known positive inotrope shown in orange, which dramatically increases contractility. Drug X does not do this. The right panel shows action potential duration measurements in the ventricular trabecula at different pacing frequencies and drug X concentrations. Again, drug X shows minimal effect on ventricular electrophysiology, confirming it does not significantly prolong the QT interval or alter ventricular conduction. Taken together with the AF efficacy data we just saw, this confirms that the drug X is atrial selective, it works where you want it to work, and it does not cause harm. Now let me transition to our lung platform. Anabios has developed a highly advanced system for testing drugs in human lung tissue using what we call precision-cut lung slices, or PCLS. The process begins with donor lung procurement. The lung is cored and then sliced using a precision instrument called a vibratom at thicknesses between 200 and 500 micron. These slices are incredibly thin, but they preserve the full 3D architecture of the lung. These precision-cut lung slices are then cultured in vitro for drug screening, or they can be cryopreserved for later use. The most important feature of our PCLS system is sustained viability. As you can see in the graph on the lower left, viability remains above 75% even at day 32 of culture. This is remarkable. It means we can run a chronic dosing experiment and disease progression studies that simply can't be done in other in vitro models. On the right, you can see that our slices are prepared with very consistent dimensions, enabling plot-based screening in a standardized format. This consistency is critical for generating reproducible results across experiments and donors. In summary, 30+ days of viability in a human lung tissue enables chronic dosing experiment and fibrosis modeling that are simply not possible with other models. Now let me show you the scientific validation of our lung fibrosis model. In this study, we used healthy donor lung PCLS, 6 lungs with 33 slices total, and we induced fibrosis by treating the tissue with TGF-beta1. TGF-beta1 is a well-established pro-fibrotic cytokine that drives collagen production and tissue remodeling. Then we tested whether nintedanib, an approved anti-fibrotic drug, could reverse or reduce this fibrosis. The left graph shows procollagen-1 alpha-1, which is a key marker of collagen production and fibrosis. You can see that TGF-beta1 treatment dramatically increases procollagen levels compared to the control, shown in the orange bar. When nintedanib is added alongside TGF-beta1, procollagen levels drop significantly. The blue bar is much lower than the orange bar. The right graph shows TIMP1, a biomarker of tissue remodeling and fibrosis. Again, TGF-beta1 increases TIMP1 and nintedanib significantly reduces it. This is a validated fibrosis model that is ready for you to test your own anti-fibrotic candidates. And TIMP1 is particularly valuable because it is a biomarker that you will also be able to track in patients during clinical trial, creating a direct translational link between your preclinical data and your clinical endpoints. In addition to our healthy lung model with TGF-beta1 induction, we have access to something very rare and scientifically powerful: lung tissue from patients who were clinically diagnosed with idiopathic pulmonary fibrosis, or IPF. IPF is a devastating and progressive lung disease with very limited treatment options. When you have IPF tissue, you do not need to induce fibrosis as in a healthy lung. The disease is already fully present in the tissue. The quality control on this slide is very elegant. You can see in the small graph on the upper right that IPF tissue already has dramatically elevated procollagen levels compared to normal lung tissue. When we added TGF-beta1 to IPF, look at the lower right graph, it does not increase procollagen further because the tissue is already maximally fibrotic. This confirms we are working with a genuinely diseased tissue. Now look at the two main graphs. The left graph shows that nerandomilast, a novel approved anti-fibrotic drug, significantly reduces procollagen levels in IPF tissue compared to the IPF control, a strong efficacy signal in a true diseased model. The middle graph tests two compounds, nintedanib, an approved IPF therapy, and drug A, a novel candidate. Nintedanib shows a strong reduction in procollagen, validating the model. Drug A shows also a detectable effect. This is a human efficacy data in real IPF lungs. You can have this data before entering clinical trials. One of the great strengths of our lung PCLS system is the ability to measure both anti-fibrotic and anti-inflammatory effects in the same tissue in a single experiment. This is clinically relevant because fibrosis and inflammation are closely intertwined in lung diseases like IPF and COPD. Fibrosis drives inflammation, and inflammation drives fibrosis. If you only treat one, the other continues to progress. The left graph shows IL-6 levels, a key inflammatory cytokine, under three conditions: the control baseline, TGF-beta1 stimulation, which increases IL-6, and TGF-beta1 with nintedanib, which reduces IL-6. So nintedanib has both anti-fibrotic and anti-inflammatory effects in human lung tissue. The right graph shows a separate inflammatory challenge using LBS, a bacterial treatment that triggers a strong inflammatory response. LPS dramatically increases IL-6 compared to the control, confirming the tissue is immunologically active and responsive. Together, these models allow you to characterize whether your compound has a dual mechanism of action, anti-fibrotic and anti-inflammatory, in the same human tissue system. This can be a very powerful differentiating feature in clinical development. Beyond functional data, our lung PCLS system also enables molecular biology studies, providing a deeper understanding of the biological differences between diseased and healthy lung tissue. On the left, you see a PCA of gene expression, similar to what we showed for cardiac tissue earlier. In this case, we are comparing lung tissue from ILD donors with normal donors. You can see that they cluster separately, confirming that diseased tissue has a fundamentally different molecular profile. On the right is a volcano plot showing differentiated expression of a gene. There are hundreds of genes that are significantly upregulated or downregulated in ILD tissue compared to the normal tissue. Each of these genes is a potential drug target. This type of analysis allows you to validate whether your target of interest is actually relevant in the human disease and whether your drug is engaging that target in the right direction. The clear separation between disease and controlled tissue ensures that the targets you identify are grounded in real patient biology and not in an artificial cell model. I want to share a real-world case study that illustrates how Anabios supports the complete drug development workflow, from target discovery all the way to efficacy confirmation. This work was done in collaboration with AbbVie, one of the world's leading pharmaceutical companies. The workflow has 4 steps, all performed in a human lung PCLS. Step 1 is target discovery. Using published single-cell RNA sequencing data from IPF lung tissue, AbbVie identified a target called target Y that is significantly upregulated in fibrotic lung cells compared to healthy cells. Step 2 is target expression validation in PCLS. AbbVie confirmed that target Y is indeed expressed at higher levels in diseased PCLS compared to non-diseased PCLS. This confirms that the target is present and relevant in a human lung disease tissue. Step 3 is target engagement in PCLS system. AbbVie tested whether a specific target Y inhibitor could actually engage and reduce target Y activity in diseased PCLS. Compared to nintedanib as a benchmark, the target Y inhibitor showed strong target engagement. Step 4 is response to treatment in PCLS. Finally, they measured whether inhibiting target Y reduced extracellular matrix deposition, the buildup of scar tissue that is the hallmark of fibrosis. The target Y inhibitor reduced ECM deposition substantially, confirming anti-fibrotic efficacy. This complete 4-step workflow, all in human lung tissue, provided AbbVie with a compelling human efficacy data for their novel target before committing to animal studies or clinical trials. Continuing with the AbbVie collaboration, let me show you one more level of sophistication in our PCLS system, single-cell analysis. Using flow cytometry, a technique that allows us to identify and count different cell types in a mixed tissue sample, we can track specific cell population within the PCLS before and after drug treatment. This slide shows UMAP plots. Each dot in these plots represent a single cell, and cells that are similar to each other cluster together. Different colors represent different cell types. The left plot shows healthy PCLS. You can see a diverse population of cell types, including T cells, B cells, epithelial cells, fibroblasts, and a small population of myofibroblasts shown in pink, which are the cells responsible for producing scar tissue in fibrosis. The right plot shows the same tissue after LPS stimulation, which activates an inflammatory response. You can see that the myofibroblast population has expanded significantly. These are the disease-driving cells. By tracking this myofibroblast population, we can identify which cell types are most affected by a drug, giving you a much more detailed understanding of mechanism of action than a simple bulk measurement would provide. This is cutting-edge, single-cell resolution analysis in a human lung tissue. Let me bring this all together with a discussion of why the industry is moving in this direction and why leading pharmaceutical companies have chosen to partner with Anabios. There are 5 key reasons. First, FDA tailwind. The FDA actively supports the reduction of animal testing. Legislation such as the FDA Modernization Act has specifically enabled the use of human-based data. Human tissue data strengthens your IND package and aligns with the direction regulators are moving. Second, cost leverage. A single phase 2 clinical trial failure costs 100 times more than a comprehensive human tissue program with Anabios. Investing in human data early is not an expense. It is an insurance policy against catastrophic clinical failure. Third, investor preference. Increasingly, venture capital investors and strategic partners prefer companies that have generated human data. It signals scientific sophistication and reduces perceived risk. Fourth, competitive differentiation. In a crowded therapeutic space, human data sets your compound apart. It tells investors, partners, and regulators that you have done everything possible to ensure your drug is ready for patients. Fifth, faster and cleaner review by regulators. Direct human pharmacology data reduces the number of questions from FDA reviewers. It builds a clearer, more compelling scientific story. The companies you see on the right-hand side of this slide, Lilly, Pfizer, AbbVie, Novartis, Sanofi, Vertex, Boehringer Ingelheim, and the FDA, have all trusted Anabios with their programs. We look forward to adding Korean biotech companies to this list. And that brings us to the end of today's presentation. Let me offer a brief summary before we open for questions. Anabios is the world's leading provider of human predictive data for drug development. We have 15 years of experience, 200 pharmaceutical partners, 50 programs advanced to IND, and a direct collaboration with the FDA. Our cardiac models, including cardioprime, ventricular trabecula, and disease-specific atrial fibrillation and heart failure models, provide the most predictive cardiac safety and efficacy data available in the preclinical space. Our lung models, including our precision-cut lung slices with over 30 days of viability, enable fibrosis modeling, inflammation studies, single-cell analysis in true human disease tissue. Together, these capabilities allow you to answer the most critical questions in drug development before you commit to clinical trials, before you invest tens of hundreds of millions of dollars, and before you expose patients to unknown risks. If you are developing a drug in any of this area we discussed today, cardiac safety, heart failure, atrial fibrillation, lung fibrosis, IPF, or COPD, I would encourage you to reach out to us. We would be very happy to discuss how Anabios can support your specific program. Thank you so much for your time and attention today. I now welcome your questions.